Hello guys, welcome to another YouTube video tutorial session on bioinformatics. Today we are going to look at genome comparison. Many a times one of the questions we want to answer with our genomes is how similar they are to a particular reference genome, for example. There are a number of ways to go about this, but our focus today will be to use the tool called BRIG, B-R-I-G, to do that analysis. The software, the full name is Blast Ring Image Generator, BRIG for short. Before you be able to use this software, you also need to have Java and then Blast installed. Okay, without these two, the software will not be able to run as it should be. So you need to do that. One good thing about this software is that you can run it in various platforms: Linux, Windows, Mac. You just need to have those dependencies installed. If you want to read more on this software, this is the paper on the software. You'll just Google BRIG and then the download link for the software will be displayed, then as well as the page to go and reach this paper on it. Okay, as the saying goes, a picture is worth 1,000 words. So I will demonstrate with an image first. This is the image that I created with this BRIG software. So you realize on the right side of it, we have some labels there. So the first one is AG199. We have other ones. So these are labels you give it during the configuration stage. My sample of interest was label MU45, but I wanted to compare with AG199, which is the reference genome for Mycobacterium ostrans. I also wanted to compare to SGL03 strain, that's also a Mycobacterium ostrans strain, then compared to Mycobacterium leaflandii and Mycobacterium schinswansii. So that's why we have this label here. You come to the center of the ring, you have AG199. So this is a label I gave it because this was the reference genome I was using. By default, the software also gives the genome size of your reference genome, so that will also be here. Okay, in terms of explaining this ring structure, that will come at a later part of this video. So let's proceed with the tutorial. I already have my software set up already, so I'll open it with Java. So first of all, I'll need to cd to where the software is. The software is here, this bring.jar. I will need Java, so I already have Java installed, so I'll use that to open the software. Great. So this is the first interface that will pop up. So the first entry here is your reference genome. So you use that to go and then select the reference genome that you want to use. So I select it. It's located here. Then I come and then I select it. The second session is for you to select the genomes that you want to do the comparison on. So I will recommend that you put all of them into a single folder or directory, such as what I've done here. Both the reference genome as well as the sequences you are interested in. So this is the reference genome. These are the sequences I'm doing the comparison on. So I browse to go and select the folder or directory where the genomes are. So this is the directory. Then I click on it. So after that, I proceed to add to the pool. Now, after doing that, just make sure you have all the genomes present. You can just go through. Then you specify the output folder. That is where the results is going to be saved to. So I use this particular directory here, folder for Windows person. Then I select once you are done, we'll click on next. When we come here you set up your ranks and then give some other customizations. Okay, so this is the first ring. Usually I prefer to use the reference genome as the first ring, so I give it a name, in this case AGY99, and then I come and add the data. The data is the file that has that particular genome, so it's AGY99 faster. Then I add it. You can also do some customizations with the color here. Okay, so you click on it, and you can use these tips here to change the colors. So we have another one here on this, or you can also use the color codes here, or you can use the RGB color codes and then change the hue saturation value as well. I will use the RGB values. So for the reference, I will use 22200. Add another rank, that's for my isolates. I add the data, change the color, 3519819. I do for the third rank, that's SGL03. I add data, change the value. We add the rank number 4, that's rank Hyundai. Add the data, change the color. We add our last rank, that's transparency. Indicate where the file is, and we change the color. Okay, let's make sure we have everything set up properly. Okay, now aside this color change, there are other customizations you can do as such as the percentage identity. By default, BRIG uses 50%. So that means that if it considers sequences as similar if it has 50% identity. There are other options you can change when you come to preferences. These options are there. You can customize them however you want it to suit your needs, but that's not the focus of this tutorial. So you can do that on your own. I will skip that particular aspect and use the default settings. Now, once we are done, we now go to next. So here we set the image title and then finally submit the work. So what we do now here is that we specify the title here in this section here. So I'll give my title and then the output file. We've already specified the folder, so the file is the name of the file that will contain the image that will be displayed. So let me say comparison file. Now you can also specify the image format that you want. It's very important that you specify it. There are different formats here. You have JPG, PNG, SVG, and then SVG. So depending on what you want, you can specify the one that you want to use for the file. Usually I prefer to use SVG because I want to edit the image later on, but for this tutorial let's just use JPG as a particular format. So once you are done with setting up with that particular aspect as well, now you can proceed to submit. Submission means now you are okay, so BRIG should do the rest of the work. So this is the key. It's before that the software also indicates the query file, just for you to make sure that the right file that you are using. So once you are done with it, we proceed to submit. Now it starts working. So depending on the genome size and the number of genomes we are doing the comparison on, the analysis will take some time before it's complete. And once it's done, you have these writings there that indicates that it's done. So once it's done, you can go into your folder. That's the folder that you want to do the analysis on. Then you can now check the outputs and then see what file was created. So there's the folder that we had. So in the folder, you can see that the file has been created. There's the file. Let me open the folder for you to check. So this the image that has been created. That's the file that we are interested in. Okay, so if you open that particular file, this is what you have. So if you look at the image here, it was JPG. Now there's the title I gave it, and then there's the genome size that was specified. Okay, and these are the labels that I was using, referring to the genomes. Now if you look at this particular ring structure here, you realize there are some gaps at certain places. Now these gaps indicate that those particular regions, the sequences there, when compared to the reference genome, they are not the same. Remember, we talked about the 50% identity threshold. So it could be that these sequences are not identical in terms of their percentage identity. So it could be lower than 50. And that is why BRIG says it's not the same. So it makes it as a gap. Or it could be that it's not really there. Okay, so this particular one tells us that there are gaps. So you can look into it more with other types of analysis, maybe looking at SNPs or some other stuffs. Okay, now aside that, you could also use this visualization to answer other questions, which maybe we'll cover probably in some other video aspect as well. Now if you look at this section here, you realize that the labels here, there's a bit of an overlap with two of the ranks here. So this is where I usually prefer to select the SVG as my format so that I could use a software called Inkscape. That's an example of a software to manually edit this image so that we have it nicely placed, just like the one I showed in the PowerPoint presentation earlier on. So with this done, I will say that will be all for this tutorial. So let's meet again sometime for another one. Goodbye.